on putative etiological and pathophysiological factors underlying ASD. John was involved in over 35 highly cited peer-reviewed articles on ASD dedicated to this work. The ACH team's most significant achievement at the time was conducting one of the only positive phase 2 clinical trials that investigated a novel intervention, which was built the first positive precision medicine initiative in neuropsychiatry. He has extensive ties to the autism clinical research and patient community and a wide knowledge base of ASD from a clinical, scientific, and business perspective. And he was at ACH, he was a personnel manager for the autism program and managed a team of 10 and was the director of autism translational research under Drs. Richard Fry. John also joined a software startup company in 2017 where he was in charge of sales and R&D. So with that, I will welcome John. Please take it away.
Thanks, Pramila. Doesn't look like, there we go. Now I can share my screen. Okay, can you see my screen okay?
Yes.
Okay, wonderful. So thank you, Pramila and the Brain Foundation so much for the opportunity to talk here today. And thank you for the kind and wonderful introduction. Today, I'm going to give some brief background on Biorosa Technologies, as well as a recently launched clinical trial that we are in process of enrolling participants for, that's entitled a Metabolic Autism Prediction Study. This study is a multi-center prospective double-blind case-controlled trial assessing specific metabolic abnormalities for prediction of autism against the current gold standard in children that are looking for a first-time diagnosis that are currently on a wait list to see a developmental pediatrician. Everyone here is probably very familiar with the autism prevalence rates and costs to society, so I won't belabor too much of this, but for those who aren't or new to the field or just trying to learn, the CDC just updated their recent autism prevalence rates just last week to show that children with autism are believed to be about 1 in 44 children in this country, which is a roughly 241% increase since the year 2000. Unfortunately, since the autism rates appear to continue to be going up, there's no FDA cleared treatments for autism. The average wait times for receiving a diagnosis continue to unfortunately lead to many delays. The cost to society is also skyrocketing as well. This graphic to the right was a recent publication by Dr. Richard Fry and a few others showing estimated costs to society and what that could mean over time for caring for children with autism and cost to the healthcare system overall. So for those who may not be familiar with how autism is diagnosed and treated today, it is diagnosed through time-consuming, expensive one-on-one evaluations between a child and a specially trained therapist. Just for context, the American Academy of Pediatrics has recommended universal autism screenings as early as 18 and 24-month well baby visits. However, due to rising autism prevalence, as well as limited number of practitioners and many other factors, unfortunately, the average age of diagnosis is still 4.5 years of age and older. And that's problematic just because the reason we really want to get children into services much earlier, just like anything in medicine with early detection, can lead to early intervention. And really the 18 to 24 month time frame could lead towards taking advantage of neuroplasticity windows that are still quite open. If we could intervene even earlier than that, great. But there's some practical reasons why that's not possible today, given the current behavioral diagnostic paradigm. To make a complicated and multi-layered problem worse, and it's just like every industry, COVID-19 really starts exposing even more holes in all areas of in every industry. But for autism, given that diagnosis is made through one-on-one evaluations between child and a therapist, the autism diagnostic test that is considered the gold standard, the ADOS. If clinicians are wearing PPE and things of that sort, the test was not validated, of course, to assess children while clinicians are wearing PPE, which makes matters complicated. And then since behavioral therapy involves one-on-one interactions to try and get children to learn different ways of changing their behavior through interactions with a therapist, since those sort of interactions didn't happen at the rates they normally would, unfortunately, children in terms of diagnosis were not being diagnosed at the rates they normally would, and the diagnoses that are happening are problematic due to the PPE, et cetera, and the therapy that children are receiving was also compromised as well. So many problems in terms of the current behavioral and treatment paradigm. So, we all are aware that, we need to try and do something to move the field forward and to try and solve many of these problems. And that's why at Biorosa, we're looking to try and develop what we hope to be a universal blood test that can be used for early detection of autism through assessing very specific metabolic abnormalities. This is not a theory or hypothesis that Biorosa developed as a company. This was something that Dr. Jill James, Richard Fry, and others that I worked with at Arkansas Children's from 2010 to 2017 helped open my eyes towards. And then really that work is still ongoing, but a lot of Jill's work that she has done over the years was retrospective. It didn't really look in terms of trying to develop a diagnostic test for autism. There was considered somewhat of a breakthrough in 2017 when Jill's data that assessed metabolic abnormalities in children with autism was provided to Jürgen Hahn at RPI in Troy, New York, in which Jürgen looked to see if he could train some computer code and develop a machine learning algorithm that could differentiate autism from non-autism cases. And this, in the studies that Jürgen did, he looked at autism versus typically developing, as well as siblings of children with autism, and saw that the computer algorithms could differentiate autism from the other groups with close to 90% accuracy. Given that was pretty remarkable, and that there was still some work that needed to be done in order to clinically validate these things and try and develop a clinical and commercial grade assay. Biorosa incorporated as a company in 2018. We had some technical challenges that we had to overcome based on some of the limitations of the academic work in terms of the laboratory techniques that were used in the academic labs wouldn't necessarily be sensitive or suitable for large-scale commercial assays. So we had to develop something, and we successfully validated our laboratory approaches. And then we also realized that given that we're dealing with metabolites, which we'll get to in a few minutes, they're involved in redox metabolism, we knew that they were highly reactive and unstable. So we had to figure out new stability protocols to try and make sure that we could not have as we use in software, a garbage in, garbage out sort of data process, and actually ensure that we have a good quality signal of the data. So Biorosa accomplished those goals, and our team innovated over the past few years, and we're fortunate to get to a point where we just recently, right before Thanksgiving, launched our prospective double-blind study, which I'll talk about here in a little bit. So what we're doing is hoping to develop what we consider what we'd hope to be a universal screening test for autism that we would hope to detect autism as early as 18 months. If we could get to a point where we could detect even earlier, great. But given that the current behavioral testing through the ADOS and other behavioral diagnostic instruments our lower bound age limit, at least starting off, in order to try and show a correlation with the gold standard, needs to start with how we can currently diagnose autism today. So we are targeting 18 months of age as our starting point. As many companies in this space or anyone that's trying to develop new biomarkers or early detection techniques for autism, we all deal with the same sort of problem. So we all start around the same age range to start. So we basically do what we do through collecting a small sample of blood from children in a standard phlebotomy setting. We are able to stabilize the blood through some proprietary ways in which Biorosa has developed stabilization techniques of our analytes of interest. The data gets shipped to our laboratory up in the Boston area, in which we've developed a liquid chromatography triple mass spec, fancy words, just a laboratory approach to being able to analyze the analytes of interest. And then those analytes, once we have our trial completed, end up going to bioinformatics to train an algorithm. And then we have a holdout set in which we validate the algorithm against our remaining set of samples. And we'll kind of talk about that a little bit later. Based on the historic research that was conducted at Arkansas Children's through Jill, Jurgen, Richard, and others, the current data has shown that the validation algorithms are performing at close to 90% accuracy in terms of differentiating autism spectrum disorder versus non-autism spectrum disorder. And that is looking at siblings as well as typically developing children. What really needs to be done and why we're doing the clinical study that we're doing is that we need to do this in a prospective double-blind study against the current gold standard. This slide just gives a little bit more context into starting with targeted metabolic analyses, looking at problems in 1-carbon folate mediated metabolism in their branch chain. That's really what Jill initially focused on, in which we saw problems in glutathione redox metabolism, problems in the methylation capacity of the cell. Folate, we've known folate super important for neurodevelopment for quite some time. And then through some of Richard's work, as well as others, we found biomarkers of mitochondrial metabolism as well that also may be abnormal in children with autism compared to other children. And this just really kind of breaks down why we need to be better at allowing for early development. You can see that where the black line is for the current age of autism diagnosis, we've really missed out on many of these key areas of neurodevelopment in which these skills are being developed by young infants. Young infants are able to develop these skills and be able to actually have significant responses to certain therapies if we're able to get children into therapies early enough. But you can see that unfortunately these windows are closing, and that if children are diagnosed here, unfortunately they've missed out on those early intervention windows in which they may have profound response to therapy. We do know that not all children have a dramatic response to behavioral intervention. And that's why we really need to not only develop and validate clinical biomarkers for early detection, but our company is hopeful that we can also leverage these biomarkers to help aid in novel therapeutics that are under development as well. But in order to get to those lofty goals, we need to start and prove out this hypothesis that redox mediated abnormalities can be predictive of autism versus not autism. And what we mean by not autism is children that are referred to a developmental pediatrics clinic for an evaluation of autism because it's believed that they may have the condition, but they're found to have other developmental problems that are not actually autism spectrum disorder, which is really the true test, right? So if we really believe that redox mediated abnormalities are predictive of autism, we need to show that it's predictive of autism spectrum and not just predictive of general developmental concerns in children. That study has never been done. And so Biorosa is the first to look at looking at redox-mediated abnormalities and 1-carbon folate related metabolic disturbances for prediction of autism. And we're very excited about the clinical trial that we just launched. And we are doing this in a prospective double-blind manner. Why? We found through our work, as well as some collaborators, that samples that we've looked at that what we consider retrospective in terms of children that had been involved in autism clinical trials post-diagnosis that were in a biobank. We see that if samples are not preserved in certain ways, they are very compromised in terms of being able to assess problems in redox. So we realized that we needed to, moving forward, develop ways in which we stabilize biomarkers of interest to where we have a good quality, clean signal that has not been compromised moving forward. For this particular study, we consider success to be differentiating autism from non-autism as defined by DSM-5 and ADOS criteria with at least 80% separation. And we're also doing, as we'll see in a few minutes, many other cognitive, behavioral, and medical assessments of the children to see if we can look at correlations with our autism risk score with other aspects of development, such as the Vineland, the Mullins, which is can look at receptive expressive language and also give a surrogate of IQ, as well as a few other psychometric tests. This just really breaks down what I've just stated in more of a graphic display. All children that come into the study from a pediatric waitlist will be assessed through gold standard criteria using ADOS, DSM-5, the Mullins, Vineland, and so on and so forth to either have a gold standard confirmed ASD or not ASD. We'll realize that those children will have another form of developmental concern, or maybe those children, the parents were worried, but the child may actually be, it may have been what we consider a worried well, where the child is actually found to be typically developing and not have any problems. Then we also have another group of typically developing children that we're recruiting at our clinic up in Vanderbilt University in Nashville, Tennessee. We define typically developing as children that have no history of developmental, behavioral, or clinical problems. The age range for the children in this study is 18 to 60 months. And so far, we have completed our first few patients that have been enrolled, and we look forward to having a few more before the holidays. And things are really picking up for what we've scheduled for January, which is pretty exciting. So in addition to all the standard clinical evaluations that are done on the children, we're also drawing a small amount of blood on the kids as well. And the goal is to see if our metabolite panel that looks at problems in redox, mitochondrial dysfunction, folate metabolism, can be predictive of autism as compared to the gold standard. And again, success for this particular pilot study is considered 80% accurate or greater. This just breaks down the schedule of events for the participants. You can see that on the left, for children that are receiving an ASD evaluation, there are two clinics. One is our site out in Arizona at the MELMED Center in collaboration with the Southwest Autism Research and Resource Center, or SARC. That is our Arizona site. And then we also have Vanderbilt University, that's also another clinical site. All children will receive the same evaluations and collect a little bit of blood. And then we also have typically developing children, a small cohort of 50 typically developing that we just verify as TD through a few different evaluations. We'll also look to see, as I said previously, whether there's correlations between some of these other tests and behavior, but that's TBD. Nothing that we do in our lab would be possible without our phenomenal scientific team. I am not a scientist or clinician, and I have much smarter people that have been able to innovate in our lab, and just truly, it's outstanding work. They've been able to achieved some very complex technical milestones that a lot of people thought before we started this company wouldn't even be possible. So the tenacity of this team and the dedication to try and develop an assay that can be predictive of autism in early life is I thank Marie, who is my co-founder and CSO, for her leadership from the laboratory side of things, and Sanjeev Biorosa is our mass spec chemist that has just done an outstanding job. We have a bunch of advisors and people that help us in many key ways in the study or for the company. And I won't go into great detail, but we've got a tremendous team that assist us across the board. Special thanks to the Brain Foundation, the families that are taking part in our clinical studies, our academic collaborators at RPI, MELMED Center, Vanderbilt, Medical Mind for their help in terms of the EDC, our investors. Personally, special thanks to Richard Fry and Jill James, who without their support of me, I wouldn't have been able to be where I am in my professional career. So I'm forever indebted to them. And for the patients that we saw at Arkansas Children's Hospital for the 7.5 years that I was there, I made a commitment to those families that I would never waver and try to do something impactful for the ASD community. And I was true to my word on that. And I thank you guys for always believing in this work that Richard and his group continues to do, but also what we're attempting to do here at Biorosa. Our children and families deserve better outcomes. We need an improved standard of care. We need better ways of trying to address the myriad set of problems that are unfortunately leaving me and the children out in the cold. So at Biorosa, we're trying to do our small part in order to improve certain aspects of a complex set of problems. And I thank you guys for your attention and for the ability to speak to you here today. So with that, I'll turn back over to Pramila.
So the next speaker is not here yet. That would be Mark. Instead.
So Pramila, did you want me to
Yes.
do the introduction? Okay. We have Dr. Stephen Oquinn joining us for our next session. Dr. Oquinn is a PharmD and senior pharmaceutical executive with over 30 years of experience in clinical development, medical affairs, and commercialization of medicines in multiple therapy areas, including neurology and psychiatry. Dr. Oquinn will be giving updates on ZOIN-002, which is a cannabidiol treatment for autism and Fragile X. And prior to joining Zynerba, Dr. Oquinn spent over 20 years at GSK and has had an extensive career in developing clinical pharmaceuticals. It's my pleasure to introduce Dr. Oquinn and look forward to your presentation.
Thank you, John. Let me share my screen as appropriate. Apologies, as it's not showing up like it should. Can you see my screen?
Yes.
Okay, it's didn't do the normal typical way, but let me put it in slide view and we'll begin. First of all, thank you very much for the invitation to present today. I've truly enjoyed what I've listened to so far, and I appreciate the Brain Foundation's efforts to pull this together to help advance the care and understanding of autism and helping families and individuals with autism. As John said, today I'm going to talk about some research that we've done at Zynerba Pharmaceuticals, specifically around ZYN-002, which is a cannabidiol treatment in a transdermal gel form. And I'll walk you through that information, tell you a little bit more about what we're doing and some of the findings we found in some of our early work, and we continue. A disclaimer here, obviously, ZN-002 is still an experimental treatment. It's not intended to promote the use of ZN-002 or cannabidiol in this instance. Obviously, we're still in clinical trials, and this information is being shared really to advance the understanding of what is going on in the field with our therapies and our products. In terms of background, obviously, this is an informed audience. You understand what autism spectrum disorder is. As you know, I'm also talking about autism spectrum disorder in the context of Fragile X syndrome as well. Fragile X syndrome is the most common monogenic cause of autism spectrum disorder. So I'll be presenting some data from both a study in autism spectrum disorder patients without Fragile X syndrome, and then also with comorbid Fragile X syndrome as a diagnosis. As many have said before me, that unfortunately, as we continue to try to press forward, there's only been 2 FDA-approved medicines for the treatment of the symptoms and core symptoms of autism spectrum disorder, and those were specifically atypical antipsychotics for the treatment of irritability. And our research is looking at what happens when you impact the endocannabinoid system with cannabidiol. And so the background for those who may not have awareness, the endocannabinoid system is a key modulator of emotion and social behavior, and is dysregulated in ASD and Fragile X syndrome. We're learning more and more every day, but for example, in Fragile X syndrome, we know that the endocannabinoid system basically is a homeostatic type system. It helps our body and brain regulate what's going on within the neurochemistry, also in development. And that system is significantly impacted because of the lack and because of the genetic challenges within Fragile X syndrome. And so we continue to explore exactly what may be happening there, but we know, again, it plays an important role. And then we know from cannabidiol, we're becoming more familiar, and probably many on this call are thinking about cannabidiol as it's been in the news a lot in the last couple of years. It is the main non-euphoric component of the cannabis plant, and it may provide therapeutic benefit in ASD and Fragile X through its effects on the endocannabinoid system. And specifically, you might wonder how is ZN-002 made, and how does it differ from what might be available currently in many states over the counter? Well, actually, ZN-002 is a pharmaceutically produced cannabidiol, so it's not plant-derived. It is actually pharmaceutically produced, and the doses that we're using in our trials are significantly greater than you would be able to typically purchase, or you'd be using a lot of CBD if you were purchased over the counter. The other important thing to know about this formulation is actually has penetration enhancers to help the absorption. If you were just to use cannabidiol itself, it would not be absorbed through your skin. And so it does require additional absorption enhancers, even if it's taken orally. So that's some things just in background for context for why we're studying the formulation that we have, ZN-002. We've done and have ongoing 4 trials that have included or will include patients that have autism spectrum disorder. I will be reviewing 2 of these in detail, but the first one on this table shows you that we call BRIGHT. It was a study specifically in autism spectrum disorder children aged 3 to 17. It was an open label trial, and I'll show you the design and the outcomes in just a moment. But we had 37 patients in that trial, and 92% of those children were at least moderate to severe ASD based upon the ADAS-2 instrument, and looking at the scoring within the ADAS-2, which many of you are familiar with. FAB-C was our first open label trial investigation in Fragile X. It was a small trial of 20 patients. We did not capture the diagnosis of autism spectrum disorder in those children, or the co-diagnosis, but we know from our ConnectFX study, which is our large randomized double-blind placebo-controlled trial in 212 patients, that in that trial, which we think would be similar to those in the FAB-C trial, 85% of the children had autism spectrum disorder co-diagnosis, and 75% of the children in the trial had moderate to severe autism spectrum disorder symptoms. And now we just initiated and started a follow-on trial to ConnectFX called Reconnect. And I'll talk about the design of that trial and what is going to be coming in the future and what we're doing with Zynerba. So first, I'd like to talk about the BRIGHT study, and I would be remiss without thanking the investigators who did this trial. This trial was run in Australia by Dr. Honey Husler and her team there in Queensland, Australia, and all the families that obviously participated and contributed to this information. The trial was designed to enroll patients with the Aberrant Behavior Checklist community score for irritability of at least 18 and a clinical global impression score, severity score of at least 4, which is moderate or greater in severity. And so we've heard about many different tools today to measure symptoms of autism spectrum disorder. And the Aberrant Behavior Checklist is a very common one. It's actually been used, and it was used in the previous trials for the approval of the 2 medicines that are approved by the US FDA. And irritability was also that endpoint used for that. And so we wanted to determine what was the outcome if we used this tool in our trials as well. The primary objectives of this trial was to look at safety and tolerability of ZN-002, both in the short term, and we looked at 14 weeks, and then we continued for another 24 weeks. So we've got 38 weeks of treatment experience, both from a safety perspective and looking at efficacy over time. And the primary efficacy assessments we looked at were, we did look at were the ABCC and the CGI improvement, which you've heard again in other presentations today. I'll talk about some other instruments we used to look at some of the core symptoms of ASD as well. We used a weight-based dose. So this is a transdermal gel. So if you imagine what that might look like, it literally looks like a ketchup, little ketchup plastic. You know, if you've gotten a takeout box with ketchup in it before, it's that you tear it off and you squeeze it out. That's basically how this is packaged, and it is weight-based dosed. And so children on the lower end of the weight would get 250 milligrams a day, and higher weights would get 500 milligrams per day. And again, the last thing on this slide is thinking about where we were looking to say, did they show significant improvement to continue in the longer term piece of the study? And we required at least a 35% improvement in the ABCC irritability scale. This is similar to some of the information you heard earlier, where you saw a 25% improvement plus a CGI score improvement. In this trial, because being open label, we wanted to raise the hurdle a little bit, and we went to a 35% improvement in irritability to make sure that we were carrying forward individuals who demonstrated benefit and could look at the long-term benefit of using ZN-002 in these patients. A little bit more about the population. As you can see, the mean age, both in those who began the trial and who continued in the second part of the study, was a little over 9 years. Similar to all the other work, majority were boys. And then in terms of race demographics, similar to what we see in standard trials. On the right-hand side, you can see just the flow of patients in terms of the first patients. We did have a couple of patients who did withdraw. Some of those were due to just the working within a clinical trial setting and some of the challenges of doing trials in children with autism. And then we looked at 28 children who completed that first 14 weeks, and 18 of those continued and met that criteria to continue on in the study for long term. This is a busy slide, but I'll set it up for you, and you'll see a second slide that's similar with a different instrument. This is the Aberrant Behavior Community Checklist. And what you see the different assessments that are made based upon the types of symptoms that are measured, with irritability, inappropriate speech, stereotypy, social withdrawal, and hyperactivity. On the left-hand side, you can see the population in period 1 of the trial, and you can see there were generally good improvements in all the domains on the ABC, and specifically for irritability, we saw about not quite a 40% improvement in irritability, and that is similar across the different symptomatologies that the ABC measures. On the right-hand side, we said, what happens in those 18 kids who continued? Did they continue to improve? Did they sustain their improvement? And what you can see here, generally, there was a little bit more improvement over time in the ABC, and that was sustained over time. So what was seen at the 14-week mark was pretty much carried forward over time through 38 weeks in those different assessments of symptoms. When we looked at some of the core symptoms using the autism impact measure, again, slightly different instrument, but looking more at core symptoms. Again, a similar picture, looking at the different things that are measured: atypical behavior, communication, peer interaction, receptive behavior, and social reciprocity. Again, you can see in general, we saw improvements on some of the core symptoms of ASD, with the largest improvement seen in atypical behavior and receptive behavior, both in the short-term 14-week period and on the right-hand side in those who did continue on. Again, a similar profile, atypical behavior and receptive behavior having the greatest change, but again, you can see in general improvements in some of the core symptoms with the open label study with Zen 002. And this is because of the nature of trying to understand what might Zen 002 do and how do children respond. We also included other things to look at. And on the left-hand side, we have the parent-rated anxiety scale for autism. And we know that through our research that part of the challenge is actually looking at anxiousness and how that actually drives other behaviors such as irritability and different findings in kids with autism. And so we wanted to look at these. Unfortunately, there's not great instruments that actually that can be filled out by the child themselves. So these do rely upon caregiver or parent rated scales. And so this is one. Actually, both of these are parents as well, as was the ABC and the AIM. But this is an anxiety rated scale, and you can see on the left-hand side, good reduction and maintained reduction in those children. And this is just in the long-term portion to make it easier versus showing more graphs. And then for the second one is actually a parent stress index, again, reducing the overall stress of the family as well. And looking at CGI, this is actually any improvement. And you see that 90% of the children are rated as having, from the clinician perspective, having some improvement. This is, you saw some presentations earlier that looked at much or very much improvement, which would be a 2 or lower score. This is a 3 or lower score. If you look at the 2 or lower score, it's about a 76% improvement. So again, in an open label nature, we found that Zen 002 seems to have promise in improving the symptoms and the parent burden in the children with autism spectrum disorder. I'll quickly summarize the findings overall, and it wouldn't be one of the key things to think about is that if you have a drug work, is it also well tolerated or not? And so that's a critical piece of any work we do. And we did find that it was generally well tolerated and consistent with other data and other trials. About half of the children and their families reported that they had an adverse event of any type, whether it was related or unrelated to Zen 02. Most of them were mild, 80% were mild. And 7 patients in the trial were deemed to have treatment-related adverse events. And I share that in the next bullet point. What type of adverse events are they? If you think about what ZN002 is, and it's, as I said, it's a transdermal gel. And all of us now have gone through the last 2 years of COVID. You're very familiar with all the alcohol gels that we put on our skins. And if you think about Purell, it's very much like that. And so because of the alcohol content, it can have some drying to the skin. And so we do see some application site reactions such as pruritus and dryness and things like that. And so that's the most common thing that we see. They tend to be mild and transient in nature. We did have one patient with sleep disorder and increased appetite and one with frequent urination. But again, overall, pretty well tolerated in the kids that we studied. And we did not find any significant changes in laboratories or EKGs, and especially looking at liver functions. That's one thing we have seen in non-transdermal forms of CBD, so it's important that we look at that. We did not see any changes in liver function laboratories and have not seen that with ZN002. So takeaways, at least from this study with all children with ASD, not with Fragile X. Again, it does provide initial evidence suggesting that when administered on top of standard of care, it seems to be a benefit in moderate severe ASD. And we did look at all the measures, and again, in the open label nature, it appears to have an impact. But because of that, obviously further trials are needed in a randomized double-blind placebo-controlled manner to confirm this data. But again, hopefully it sets the groundwork for understanding how best to use and how to study ZN002 and ASD. I want to transition now to looking at some work in Fragile X. As many of you know, again, Fragile X is the most common monogenic cause of ASD, and it's being used and has been used as a model to understand things over time with ASD. Obviously, these children tend to have more intellectual disability and may have other comorbidities, but it's a helpful way to understand what might also be happening in ASD. So I want to present some data from that and talk about some of our other work. The ConnectFX study, which was a double-blind study in Fragile X patients. Here you see the trial design. It was a 14-week trial to evaluate the efficacy and safety of ZN002, again in children, same age group, 3 to 17 years of age. And you can see using doses of 250 milligrams or 500 milligrams a day versus placebo. And again, in this trial, this was a double-blind placebo-controlled trial. And so the children who received placebo got identically matched treatment in terms of look, feel, smell, everything like that. And so it was a fully blinded trial to both the caregivers, investigators, and the children. And then they had the opportunity to go on into an open label trial that's still ongoing, and I'll share some early data from that trial as well. What we did learn is that in our primary population up front, ZN002 did not statistically separate in terms of our primary and key secondary endpoints. But one thing we did do, we had scientific evidence that said, we know that in a trial that was done by Novartis and Fragile X several years ago, there was work looking at what happens if the children have significant methylation of their FMR1 gene. And so we did a pre-planned, before we broke the blind, analysis of patients that had at least 90% methylation of their FMR1 gene. And these results suggest that ZN002 may be beneficial in that group, and I'll walk you through some of that data. We also found a very similar tolerability profile as we did in the general ASD population study I talked about in the BRIGHT study. And again, application site pain was the common adverse event when 6.4% of patients and 1% of placebo. So again, pretty well tolerated and mild and transient application site pain reaction. So let's talk about the data, because I think we're trying to understand, again, what's going on scientifically within the responsive profile, where ZIN-002 may be beneficial and should be studied. This slide actually looks at, in this point, the primary endpoint being the ABCC, actually a Fragile X scoring of that instrument, looking at social avoidance, which was the primary endpoint in the trial. And so, as I said, the left-hand group of bars, where it says full analysis set on the left, are all patients. And so you can see while we saw an improvement, it did not reach significant difference from baseline in terms of the reduction or improvement in symptoms of social avoidance. When we did the pre-specified ad hoc analysis, looking at greater than 90%, we actually did find a difference here. And you can see greater separation and a greater reduction in social avoidance scores. And we also did a post hoc looking at 100% methylation. What we think may be going on here is that, specifically in Fragile X children, you have silencing of that FMR1 gene, and they are children that are likely most impacted in the function of the endocannabinoid system. So they may be the population that would be more likely to respond to ZIN-002 or cannabidiol treatment. We will continue to explore this, but again, from a scientific standpoint and looking at what may be happening there, there's plausible explanations, but we will be repeating this work to confirm it. Another important thing that we found is not only did we see improvements in social avoidance in those groups that are highly methylated, we also looked and said, is this data meaningful? And so we did some psychometric work, and we looked at 3 areas, specifically social avoidance, irritability, and socially unresponsive or lethargic, based upon the aberrant behavior checklist, community checklist. And so what we did is we did market research with families and tried to understand what they felt were important changes and meaningful changes. And then we did this using standard methods that the FDA asked companies to look at to define meaningful change. That's what the clinical outcomes and assessment group at FDA really wants companies to do and developers of medicines to do, because just having something statistically significant is great, but is it meaningful? And so we were actually able to show that a 3-point change in social avoidance was meaningful, a 9-point change in irritability was meaningful, and a 5-point change in socially unresponsive lethargic measures were meaningful. And if you look at the data and said what percent of patients actually achieved those results, nearly 60% of children with significant methylation in our trial did so, and more so statistically than placebo. Irritability, again, while it didn't separate in the general group, it did do when you looked at those who had meaningful change. And there was a positive trend in socially unresponsive lethargic, but it did not reach significant difference in that case. But again, positive and supportive information to help us have confidence in moving forward in our development. So some lessons learned from our first trial. Again, we did see a treatment response that appeared to be greatest in those with higher methylation. We were able to look at psychometric assessments and determine that meaningful change, which I just talked about with you. I didn't show you data, but we also looked at a caregiver global impression of severity and change instrument, and we were able to find that instrument actually performed pretty well as well, and have carried that forward in our next trial. And based upon our interactions with the FDA, we have looked at the clinical global impression of severity and improvement scale, and based upon their guidance, we are now going forward in our new trial, which I'll talk about Reconnect in a moment, with a more disease-specific clinical global impression assessment, looking at those specific behaviors and trying to get the best assessment possible from a clinician standpoint. And so that will be new in our trial. And last but not least, some others have mentioned just some learnings having to do clinical research during the era of COVID and the restrictions. And we've been able to move forward with a significant amount of virtual visits in our new trial. In fact, half of the visits in the new trial will be virtual, which significantly reduces burden for the families who participate and their children. Before I move forward to introduce our new trial, I did want to pause and just share some new data with you as well. And as we develop our medicines, obviously understanding the safety both in the short term and the long term is critical. Here is some data we've just found in our long-term open label trial. It's brand new data. And I probably didn't do a great job setting this up earlier, but as I said, those who participated in our blinded trial can actually come into this trial and continue therapy. The patients who were in our FAB-C original trial, you'll see 10 of those patients rolled in, and also the patients in our future trial, our new trial will be able to roll in. But as of May 21st, we had 240 children in this database, and the median length of treatment was 21 months. And so through that period of time, again, we've continued to see a good tolerability profile with no changes in vital signs, ECGs, or laboratories. And on the right-hand side, you can see, again, with long-term therapy, generally mild to moderate adverse events when they did occur, but you can see here that most of them are pretty well tolerated with things that you might expect to see in a population of children over time. And then in terms of treatment-related adverse events, again, the same thing that we saw in our blinded trial, the most common being application site pain at 6.6% of the children. So again, encouraging information to help us think about and carry forward in our development program in the long term. We also looked at what happened over time. And so the patients that were in our double-blind ConnectFX trial could also, again, roll over and continue. And half of those patients, remember, were actually on placebo, and half of them were on ZN-002. So on the left-hand side of this graph, you see the line with the squares and the line with the circle. The line with the square is ZN-002 treated patients that were blinded, but they were being treated with ZN-002, and they obviously continued on ZN-002 in an open label fashion. The bottom line with circles actually are the placebo treated patients. And then when they went into the open label trial, what happened? Here, because again, trying to understand what benefit might we be seeing with these children, we put a pretty significant hurdle in place to say, how many of those children actually had, again, the definition of meaningful change in social avoidance. And so all these children had to have at least a change of 3 points on their score for social avoidance. And we added additional criteria that they had to hold that for at least 2 consecutive visits. And so you can see that within the double-blind trial, separation from placebo. And then again, hopefully you would expect to see that once patients received, who were on placebo and then received ZN-002, would they improve, and would they improve similar to those children already on ZN-002? And you can see that they did. So again, additional information that encourages us to continue forward and advancement of our programs with ZN-002. I'll close with this slide and one final summary slide, but this is our Reconnect trial. We've just implemented this trial. Again, this is in a Fragile X population, which we expect, again, about 75% or more of the kids will likely have a comorbid diagnosis of ASD. We're looking to enroll about 200 patients, and we will be testing that hypothesis around the impact of methylation. It'll be important to understand, is that replicable? And the FDA has asked us to confirm that in this trial. So we definitely will be doing that. We've also introduced another dose level to make sure there's consistency in a milligram per kilogram basis for the larger kids that will enroll, or more heavier weighted children and older children. So we've introduced a 750 milligram dose. And again, this will be an 18-week treatment trial. We've extended the duration as we believe we'll be able to, again, show continued benefit of ZN-002 with longer therapy. The endpoint, again, is social avoidance over time, and we'll look at irritability, we'll look at social interactions, and we'll look at the clinical global impression as well for improvement to have that clinician rated score. And again, we are testing the hypothesis around the methylation perspective. Again, our prior data shows a pretty strong signal that methylation is important, but frankly, we don't want to not treat anybody else, not leave children out who could benefit as well. So again, we are going to retest that hypothesis to determine if children with partial methylation may also continue to benefit or not. And so we'll answer that question in our trial. And in conclusions for our time today, again, hypothesis generating on the impact of methylation. It does appear that these children are the ones who actually have silencing of the gene, and that may be important, at least for Fragile X. We'll continue to think about mechanistic issues for autism in general. We talked about the outcomes in terms of efficacy and safety. And again, we are actively enrolling the Reconnect trial. And so if you have families or others who have children who also have Fragile X as a diagnosis, there is information available. The trial is enrolling now if those children may benefit or would like to participate as families within this trial. So John, I'll stop there and would be happy to answer any questions depending upon our time in the chat here.
Great. Yeah, so I'll just read off a couple questions that have come through. One, was the baseline severity of children that were 90% methylated, was their baseline severity profile worse? Was that investigated at all?
So in the data we have, obviously we didn't do it prospectively, but just looking at the demographics, there were some signals of potentially more severe Fragile X syndrome. And we looked at both IQ, which was a little bit worse in that group. We looked at autism spectrum disorder symptoms. They were more similar, slightly worse. And so we'll continue to look at that in our ongoing study as well. Great question.
Okay. A couple more for ZYN002. For the non-responders, did you guys do any sort of genotyping or looking at whole exome or anything like that to see if you could find some sort of genetic reason for why there was a non-response?
No, we have not yet. We did, again, looked at the methylation profile, and I didn't present the data here, but again, what we saw was a pretty significant drop-off in response for those children that had less than 90% methylation. What we found was about 80% of in the children in the Connect study actually were 90 or above percent methylation. And so that in that 20% of kids who were less than 90%, there was a pretty significant drop-off in responses in 002. So we, again, going forward, it is an area of investigation, and we will be looking to understand that better and to reconfirm that in our Reconnect study.
Great. And just one last question, just when you're talking about meaningful change, were you referring to the minimal clinical important difference, or the MCID on the scales?
It's a very similar analysis, yes, John. Actually, it's FDA has moved from a patient and a caregiver rated assessment to meaningful within patient change. Mm-hmm. And so it's very similar to the MCID that would be with a clinical assessment, such as a CGI or something like that. And it is done prospectively to define what might be meaningful within the trial, and then you assess it within your population. And so that's what we have defined in our ConnectFX, and we'll be repeating that as well in our new study.
Did that also include, sorry, just out of curiosity, in terms of just clinical meaningfulness, but also did it also interrogate, either a parent or caregiver in terms of their perspective in terms of whether it was meaningful to them?
So in the ConnectFX trial, it was done up front in terms of defining what would be meaningful to them. In the new trial, we'll be doing post interviews, again, to understand that even more robustly. So great question.
Wonderful. All right, well, thank you so much for your time, and we'll move on to the next speaker, Mark Smith.
Thank you, John.
Which is a company developing novel microbiome drugs to serve patients with serious unmet needs. Mark has led much of the seminal work on engineering the human microbiome, publishing in leading journals such as Nature and New England Journal of Medicine. He translated microbiota transplantation from a topic of research into clinical practice by founding Open Biome, where he pioneered the universal donor model as a safe, effective, and cost-efficient solution for the treatment of Clostridium difficile, the epidemic of Clostridium difficile infection. Open Biome has now treated more than 55,000 patients through a network of over 1,200 hospitals. And Mark co-founded Finch to develop and scale a new generation of microbiome therapeutics. Mark graduated from Princeton University with a bachelor's in biology and received his PhD in microbiology from MIT. So without further ado, it's my pleasure to introduce Mark Smith.
Hey, John, thank you so much for having me here. I'm really excited to have the opportunity to share some updates on our work and a little bit more background on the microbiome and its potential linkages to autism. So I really appreciate the opportunity to share our work here today. So I will be making a few forward-looking statements as a publicly traded company. I need to share a few disclosures. Feel free to check out our website where we have a more fulsome set of disclosures available. So without further ado, I'll jump into the meat here. So as a little bit of context on the microbiome, we all have within us in our human genome about 25,000 genes. There are more than 20 million in our microbiome. So this is the community of tens of trillions of microbes that live on and inside of us. And this enormous genetic repertoire is now increasingly being understood through the application of high throughput sequencing techniques and analytical tools to really understand what this community is doing and the roles that it may have in our health. And what we've realized is that the microbiome has a really important role in modulating our immune system, our metabolism, and potentially even the function of our neurological system. And so at Finch, our objective is to really leverage some of these exciting tools from genomics and data science to come together to develop a sort of new class of therapies based on living microbial medicines. And so just a little bit of context on myself and my journey into to this space. I actually started off as an academic researcher studying the human microbiome. My wife's cousin ended up with recurrent C. difficile about a decade ago, and it was a very, very challenging illness for him. He had been sick for about a year and a half. He had failed 18 rounds of, he had failed 7 rounds of vancomycin over that 18-month period. And, for me, kind of watching his journey, I think there's some probably analogs to, what I understand a lot of families in this community feel, where there just really weren't good options available to him. He ended up sort of taking matters into his own hands, providing an at-home microbiota transplant, and worked for him. But I just felt this is not a sustainable solution. This is not how we should be serving patients. And so I started an organization, as John mentioned, called Open Biome, to serve this community and make these kinds of treatments available for patients that had failed, all available therapies for treating recurrent C. difficile. I'm really proud of the impact that we've been able to have in the C. difficile patient population. And a little over half a decade ago, I started Finch, with the objective of really scaling this sort of new platform of microbial medicines to other therapeutic areas, and to develop a new generation of products that can really aim to build effective microbial therapies that can scale and really work for patients. And so a little bit about how we do that at Finch. You'll see if you jump on clinicaltrials.gov and actually just look up all the ongoing studies in microbiota transplantation, there's really been an explosion in research activity over the last decade, where now that you'll see more than 300 ongoing clinical trials exploring microbiota transplantation, a wide range of therapies. And our own view is not all of these are going to work, but those that do, deserve follow-up. And we have just an extraordinary opportunity to mine those clinical data to identify, what are the specific organisms and metabolic pathways that are actually driving the effects that we see in patients. And we think this is a really logical approach to address this sort of new therapeutic modality, where we can really learn from what's happening in the clinic, what patients are experiencing, and go through a process of what we call human first discovery. So reverse translation from what we're already seeing in the clinic to inform the kinds of therapies that we develop to scale in order to serve patients. And so we've been very successful in applying this approach to a number of therapeutic areas. So, we have a phase 3 program in recurrent C. difficile, having completed a positive placebo-controlled randomized phase 2 study just a couple of years ago. We have a great collaboration with Takeda focused on ulcerative colitis and Crohn's disease. And then the main focus of our time today, of course, will be on the work that we're doing in autism with FIN211. And we also have a program focused on chronic hepatitis B infections as well. And so just a little bit about the tools that we've developed at Finch in order to address diseases that we believe may be linked to the microbiome. We have, on one hand, an approach that we call complete consortia. So all of us have living within us this community of tens of trillions of microbes. And what we're able to do is identify rigorously screened healthy donors and then harvest intact microbial communities from these donors, encapsulate them in enteric-coated capsules that we release at the appropriate location within a patient's gastrointestinal tract, and then establish a sort of new living community in these patients. And so on one hand, we have these complete consortia donor-derived products. And then we also have what we call our targeted consortia products. So where we've identified specific bacteria that we believe can have a big impact on patients, and then have isolated those, grown those up in pure culture, the way you might ferment beer or something like that, right? So these large anaerobic fermentation chambers. And then, again, encapsulated those in enteric-coated capsules that will release in the appropriate location in the GI tract. And what we're doing in autism with the FIN211 program is actually combining both elements of this platform. So developing what we call an enriched consortia product, which has both a complete consortia scaffold, leveraging a lot of the exciting data that we've seen from microbiota transplantation, while also including an individual strain that's not found in all donors that we believe may be really important for serving these patients. And so we'll talk a little bit more about the FIN211 program as we go, but that's the conceptual framework that we're using for the design and development of this product. And so for this community, I don't need to share how urgent the need is here. There's a really large unmet need. What I will highlight is, our development is specifically focused on the subset of children with autism that also suffer from significant GI symptoms. And we believe there are about a third of patients with autism that suffer from significant GI symptoms. We're specifically focused on those with significant constipation. And we believe that by focusing on this subset of patients, we can manage some of the heterogeneity that exists within the autism community and find a subgroup that we think is maybe more likely to respond to this therapy as an initial target for development, with opportunities to potentially expand the addressable population over time. And so, a little bit about what's driving our interest in this area and our decision to pursue a novel therapeutic based on the microbiome to serve the autism community. So, first, just a word on the epidemiology. We know that there are certain early childhood events that seem to correlate with the risk of developing autism later in life. So, whether it's C-section birth or duration of breastfeeding or exposure to antibiotics, there are a number of elements that seem to be related to the composition of the microbiome that are epidemiologically associated with the risk of developing autism. And the antibiotics data, I think, is particularly interesting because we've seen a dose-dependent effect where individuals that have been exposed to more frequent doses of antibiotics during the first 2 years of life are at a significantly higher risk of developing autism later in life. So this kind of epidemiological framework is one clue to us that the microbiome may be one important element of the pathophysiology of autism. We've also spent a lot of time trying to understand why this might be the case. And one mechanism that we find interesting is the role of oxytocin signaling in autism. There's been a lot of work focused on delivering exogenous oxytocin, given the important roles that oxytocin plays in modulating sociality and bonding. What's particularly interesting to us about oxytocin is, it may be possible to induce endogenous production, so the production within an individual by modulating the microbiome. And we'll talk a little bit more about that. Another mechanism that we believe could be very important and could be relevant both for some of the behavioral as well as gastrointestinal symptoms that children with autism experience is the role of the gut barrier function in the etiology of autism. So we know that children with autism tend to have a less robust gut barrier. So they have sort of a leaky gut, which leads to the translocation of certain neuroactive microbial metabolites, like 4-EPS is one reasonably well-known example of these. And we know that microbial interventions can help to restore the integrity of the gut barrier. And this could be one way that we both improve some of the gastrointestinal symptoms and also avoid the circulation of some of these neuroactive microbial metabolites, and also potentially influence the neuroinflammation, which we believe could also be an important driver of some of the symptoms that are experienced by children with autism. And so I wouldn't, of course, be talking about any of this if it weren't for the clinical data that supports this. Again, our whole model for developing new therapies here at Finch is to really learn from what we're seeing in patients and in the clinic. And in this case, we're really excited that we've seen data from more than 200 patients that have been treated with microbiota transplantation across a range of studies. And each one of these studies has their own limitations that I think are important to highlight, and we'll discuss some of those as we dig into the data a little bit more. But taken as a whole, we find that this is really exciting proof of principle data that guides our development and really informs our understanding that the microbiome may play an important role in the development of some of the symptoms of autism. And what we've seen is, across these studies, we've seen improvements in both GI and behavioral endpoints, again, across a number of different investigational sites, different centers, really all around the world. And there's a recent Cell paper around the role of the microbiome and autism that highlighted, I think, the importance of this interventional data. The study was evaluating the correlations between the microbiome and autism, and found that they may actually be driven by an underlying correlation with diet in children with autism. And this is exactly why we've been so focused on interventional data. So looking at what happens when you sort of change the composition of an individual's microbiome and the impact that has on their symptoms and clinical outcomes. And this is why we believe that this reverse translation approach that we're using at Finch is such an important tool for really unpacking the role of the microbiome in this population and others as well. So now we'll dig into the data together. So I'll begin by walking through one of the first studies that came out in this space. run by Jim Adams at Arizona State University. And this was published in a series of papers starting in 2017. And in this study, it was a small open label study with the caveats that come with that methodology that conducted a single investigational center. And what they did is they provided a really robust series of microbiota transplantations over a period of an 8-week sort of induction process. And they evaluated these children both at baseline, 8 weeks after the cessation of therapy, and then even 2 years after these patients were last treated with a microbiota transplantation. And what they found was really encouraging. So in that 8-week time point, they already saw statistically significant improvements in GI symptoms. Those are preserved even 2 years off therapy. But the behavioral endpoints, I think, are the data that we found most encouraging. So, again, a statistically significant improvement at the 8-week time point. And at 2 years after treatment, a number of kids, sort of fell below what a threshold that has historically been used for identifying autism. And we found that these were really encouraging. Nonetheless, we also said, well, this is an open label study, and I think there are some important caveats with that. And so, another data set that we've looked at to really inform our perspective on the potential role of microbial therapies in the treatment of autism is a study that used a comparator arm. So in this study, patients either received just behavioral therapy alone or behavioral therapy plus microbiota transplantation. And what we saw in this study is that these children again experienced significant improvements in GI symptoms and behavioral symptoms, and those were associated with changes in the microbiome, with increases in microbial diversity. And what's particularly interesting about this study and the next that I'll highlight is in these studies they found without the same really intensive induction therapy that was used in the Jim Adams study, they found that the effects seemed to wane with time. So there was an initial improvement, and then over time, some of the benefits seemed to wane. And this may inform the need for long-term maintenance therapy in order to maintain the benefits that are initially observed. And I think that's a really important question that we look forward to digging into in further detail. And the third study that I'll briefly highlight here, in part because it's just come out quite recently, just a few weeks ago, really builds on some of the other studies that I mentioned. In this study, children received either colonoscopically administered material or an oral therapy. And we thought this was important to evaluate the potential role of treatment modality. And when we found that there were no differences between those two modes of delivery, which is really encouraging for us because we believe that a colonoscopic intervention would be really challenging for children, but an oral therapy obviously has a lot of benefits in terms of access. And so these in this study, patients received a weekly dose of therapy for a 4-week induction period, and then were evaluated at 4 and 8 weeks after therapy. And so you can see that the GI symptoms improved quite significantly. And here we're showing the data around the Bristol Stool Scale, which is nice because it's a somewhat objective measure looking at the physical characteristics of stool samples that these children are passing. And at the end of therapy and even 8 weeks off therapy, there's some really significant improvements here in terms of the fraction of bowel movements that were abnormal according to the Bristol Stool Scale. Also really encouraged by the behavioral outcomes, and here we're using the CARS scores as in the earlier studies that I mentioned. And you can see the sort of temporal effects here, where there was a significant improvement at actually all of the time points, but that was most acute at the first time point, and there was perhaps some directional reversion that occurred in some of the later time points here. So, natural question again, getting back to some of the mechanistic concepts that I introduced earlier is, how does this work? And, I think there are a number of important caveats that are worth highlighting anytime we speak about, developing animal models of such a complex phenotype like autism. Nonetheless, I think these can be important tools to really try to understand mechanism because of the limitations of the kinds of work that we can do in clinical studies. And so in this animal model, where some of the key endpoints that were evaluated were the amount of time that these animals spent interacting with pairwise interactions with other mice, we found that interestingly, there's an individual strain that we knew was associated with induction of endogenous oxytocin, that when it was added, so the black here are neurotypical models, the red are the autism model, and then blue is the autism model treated with this oxytocin-inducing strain. And you can see that in this case we were able to restore neurotypical levels of these pairwise interactions by delivering this oxytocin-inducing strain. What's interesting is that was correlated with an improvement in restoration of neurotypical oxytocin levels. So, by itself, I think this would be difficult to draw any strong inferences from it. What's particularly helpful here is actually seeing some of the knockout work that was done as a follow-up to this. So if you vagotomize these mice and sever the vagal nerve, which connects the gastrointestinal tract to the central nervous system, you eliminate that rescue effect of adding in this oxytocin-inducing strain. So you can see that the interaction times are significantly lower among those mice that were treated with this oxytocin-inducing strain that had a severed vagal nerve. And sort of complementing that data, I found that if you knock out the oxytocin receptor, the gene that is responsible for the protein that binds oxytocin receptor, oxytocin, you can see that also eliminates the rescue effect of adding in this oxytocin-inducing strain. And so collectively, one of the inferences that we've drawn from these data is that oxytocin may be an important driver of in this model system, and that there are individual bacteria that we could deliver that are capable of restoring a neurotypical behavior, and that was associated with increases in oxytocin, and it appears to be induced through a stimulation of the oxytocin receptor through a signal that is being transduced via the vagal nerve. So these are all we believe important early signals that may suggest a role for these oxytocin-inducing strains as part of a broader microbial therapy that could be used to try to replicate and build on some of the results that we've seen with whole community microbiota transplantation. And so this has heavily informed our product strategy here at Finch. We've chosen to deliver what we call an enriched consortia product, as I mentioned earlier. That's the design of Fin211. And the idea is we have this complete consortia, which aims to really deliver an entire community, replicating some of the exciting data that we've seen with microbiota transplantation and this individual oxytocin-inducing strain, and sort of the ability to deliver both elements of that. And we've had some really encouraging engagements with the FDA. In our pre-IND meeting, they agreed with us that we can go directly into children with autism. And in the Aspire trial that's going to be starting in the first half of next year, we're going to be able to do just that. We're going to be able to jump right in to start serving children with autism. And they also agreed with us that demonstrating benefit for either GI or behavioral symptoms could be sufficient to support a BLA. And we think that opens up a number of interesting development strategies for us in terms of how we may be able to advance this therapy to be available and accessible to patients in the future. So I'm very excited to introduce the Aspire trial here today. So this is a phase 1b study that's designed to really help us understand the optimal dosing level and dosing regimen that we can use as we carry this product forward into later stages of development. And so the part A, the first component of this study, is going to be a dose escalation study. So patients will either receive a low or a higher dose of FIN-211 for a 2-week period. And then we'll take the highest tolerated dose from the first phase of this study into a small randomized study where patients will get randomized to either receive antibiotic pretreatment, with the idea that by, similar to stem cell therapy, where you sort of want to knock out the incumbent community in order to create a blank canvas on which you can sort of paint this new ecosystem, using antibiotics as a tool to standardize the baseline microbiome of children that are receiving this therapy, or patients with that don't receive antibiotic pretreatment. And our preference, if we can avoid the need for antibiotics, would be to move forward with a simpler therapy. We want to rigorously test that question in this first set of studies, so that we have the right both dosing level and regimen, understanding whether we need antibiotics before we move into a larger placebo-controlled study. And the primary objectives will be looking at safety and tolerability, as well as the pharmacokinetics, so how these organisms are engrafting in patients. And we'll also be measuring as exploratory endpoints some of the key behavioral and GI endpoints that we discussed earlier. And so, really excited about the opportunity to begin evaluating these novel therapies in patients next year, and to start to get some early readouts from these trials in order to understand whether we're on the right track and we're going to be able to continue to advance these therapies. So I really appreciate the opportunity to come here again this year and share some updates on where we are. I hope that next year, when we present again, we'll have some early data from these studies. And really thankful to everyone in the community that's come together to help inform the design of this study. And I hope to continue to be able to work with the community to evaluate and understand and ultimately develop some really exciting novel therapies here. So thanks, and happy to take any questions.
Thanks, Mark. There's a few questions that have come in. So one, is the oxytocin producing strain named in the paper that you were citing?
Yes, it is. It's in the paper that's cited there. They use Lactobacillus strain.
Great. Was that Reuteri?
Yes, they used an L. reuteri strain in that study.
Great. Was that the same paper that showed a connection with BH4 metabolism as well?
It's the paper that I cited is a different paper, but by the same group.
Okay. One question for you in your trial. I know it's early days and maybe not a part of this study, but are you thinking about also doing any type of neuroimaging or anything of that sort to try and look at the actual, fMRI or some other sort of brain-based changes as a result of FMT treatment?
Yeah, it's a topic that we spent a lot of time discussing with the team, and kind of weighing some of the pros and cons. We want to minimize the burden on kids and their families, but we're also really interested in that data. And so I don't know that we've made a final decision on whether we're going to include those in at some point in the study, potentially as a sub study. But the expectation is that won't be a requirement for the study at this point, but it's a topic that we're continuing to discuss.
Cool. One thing that might be interesting, if the microbiome, oxytocin, BH4 side of the story pans out in humans, Richard would be a good person to talk to. One of the studies that we published, Arkansas Children's, was actually on tetrahydrobiopterin or BH4 treatment, Kuvan for autism. We showed that there appeared to be a connection with nitric oxide metabolism, and we theorized that maybe it had something to do with improving cerebral blood flow. So be curious if FMT treatment may actually have effects on nitric oxide metabolism, and if that actually could be shown up if you do decide to do any fMRI type studies and see changes in cerebral blood flow patterns. Might be neat. So a couple other questions have come through. If the study shows that vancomycin pretreatment is important for effectiveness, is it possible to extend the same to placebo group as well, since they may be left out of the FMT treatment protocol?
Yeah, it's a great question. So in this study, Aspire Part B, there won't be a placebo group. So actually everybody's gonna have access to therapy. The group that's only receiving 10-11 won't get the vancomycin therapy as part of it, because there could be efficacy just from the vancomycin component, which is one of the questions that we want to understand as part of the study. But there won't be a vancomycin standalone arm in this study.
Okay. Another question that's come through, and I think this will be the last question before we move on to the next presenters. Do you have a theory or any rationale for why there might be a need for multiple FMTs for children with ASD? Following to that question, maybe an underlying mechanism affecting the microbiome that requires certain bacteria to be constantly replenished. Have you thought about that? And do you have any suggestions there?
Yeah, so it's a great and actually very complex series of questions around this sort of ecological dynamics and sort of longevity of these communities. The short answer is, children with autism, they have if there are sort of disruptions to their microbiome, it may be driven by, dietary preferences or other, elements that they experience. And so some, whether it's, genetics or their environment or diet, something has about those confluence of factors have selected for a dysbiotic microbiome. And so, part of our thinking is that without constantly sort of replenishing this, some of those missing microbes, they may be lost over time as they have been potentially by some of those factors. And so that could be one reason. And it's something we're really excited to be able to study here, is we'll be able to actually measure how the community changes over time and actually use the sort of community decay in part as, to inform our understanding of how long dosing may be necessary for in the future. So that's how we think about engraftment and pharmacokinetics in this context. And it's a really important and complex part of how we think about developing these kinds of therapies in autism and in a range of other applications as well.
Great. Then one last quick question, whether it be related, just in general, it could be related to autism or just, in general in terms of FMT treatment. Curious, do you know of any reports on really any condition of doing a microbiome transplant therapy and that affecting dietary preferences after the fact?
I don't know of any rigorous studies of the question, though anecdotally, I've heard of some pretty significant changes in dietary preferences among C. diff patients, though those are kind of to be expected because they're, have very, very severe diarrhea and are, often experience significant weight loss and then, have their appetites restored once the probably the underlying driver of that is resolution of their C. diff and the underlying diarrhea. But in a kind of more normal population, I haven't heard examples of that yet.
Great. All right, Mark. Well, it was always a pleasure. Thanks for joining us. And there might be some more questions. If you wouldn't mind looking at the Q&A and the chat function and get back to folks, I'd be greatly appreciated.
Yeah, absolutely. I'll do that offline right now and let you guys continue the exciting program.
Thanks, Mark. Okay, for the treatment of core symptoms of autism, Dr. Margarian, or I guess Peter Griffin, for this presentation, has over 20 years designing and executing clinical trials for multiple pharmaceutical companies. Dr. Margarian has an MD and a PhD from Northwestern University, but he also did fellowships at Boston Children's Hospital with Mass General and Harvard. Chuck Bramlage is the CEO of Yamo Pharmaceuticals and is an experienced drug developer and has over 28 products that have been launched in 15 therapeutic areas to his name. It is a privilege to introduce both of these gentlemen, and I will leave it to you guys to dive in. Thank you.
Great. Thank you for the introduction. I'll start and just give a couple of slide introduction to Yamo. I joined in 2017, very interested. A senior scientist at AstraZeneca told me about this compound, L179, and he knows I like startup companies and asked me to talk to the inventor and the investors that had done one previous trial and were in the middle of a second trial. We completed that. Dr. Tom McGarrian, our chief medical officer, joined us at the end of 17. And we, of course, along with COVID and many other clinical operations people, were not able to do anything the last 2 years. We also had an issue with raising money. We're very excited to present the current data within the company, as well as what we plan to do right now as we start our third trial. We're very excited about it. We're interested in your feedback at the end of this. And of course, our expert Tom will walk you through the science behind it. We are seeking an additional $1 million right now of investment. And that investment would cover some more patients above and beyond the 50 that we're going for now to get 40 at the end. We think we can do another 20 to 25 patients with another $1 million. So those are the three things I wanted to leave you with at the beginning. The first slide here, we are a clinical stage company that's developing L179 to treat the core symptoms of ASD. We hold IP that goes to 2033 and other indications. We think this drug may work in post-traumatic stress disorder, type 2 diabetes, and fibromyalgia. It is a racemic mixture of alpha-methylparatyrosine, which decreases catecholamine synthesis. In 2018, we had a fast track designation from the FDA based on our previous 2 trials. We didn't have enough data to get the next designation, but we feel this current trial that we're starting right now is going to give us a lot more quality data from 5 top centers in the country with 5 experts in autism along with Tom. We're very excited to see what the results will provide. Next slide, please. I'm just hitting some of the highlights here. This month, we're starting this 50-patient double-blind study. We're targeting 40 completers in this. We're having a chronic dosing of 12-week with placebo and/or a 2-period crossover with drug. These patients are 12 to 21 years of age with IQ of at least 70 and moderate impairments in socialization. These patients will be randomized between active, the 200 milligram BID, and 300 and placebo. We expect to get this information, the data completed by 2023 at the beginning of the year. We had just recently announced that Autism Impact Fund has become our first institutional investor. We've had all the rest of the investors have been high net worth individuals who have contributed and have an interest in doing something for these patients and their families. So we are seeking, again, another $2 million to cover some upside. $1 million will cover an additional 20 patients, and that's what we're working on now. Next slide. Okay, so here I would like Tom to describe the science behind L179, give you a summary of the first 2 trials very quickly, and more or less go quickly into the current trial that we're starting right now in 5 centers around the country. So Tom?
Thanks, Chuck. Can you guys hear me okay? My volume coming through, Chuck?
Yes.
Yep.
Yeah, thanks, guys. Appreciate the opportunity to talk to you about our exciting compound and what we think has some very good promise based on some preliminary data in an open label study and a phase 2A study. So alpha-methylparatyrosine is a competitive inhibitor of tyrosine hydroxylase, and what it does is to decrease the presynaptic release and stores of basically all the catecholaminergic neurotransmitters, which of course, as we know, are involved in many different functions, many of which overlap with some of the deficits seen in autism: memory, reward, attention, focus, communication, pain, self-mutilation, repetitive behaviors. There's also a lot of indirect effects that the catecholaminergic system has, including co-release of growth factors, modulation of serotonin, acetylcholinergic, vasopressin, oxytocin, and other neurotransmitters. And we believe that this is a mechanism that works centrally as well as peripherally and might address some of the other medical and problematic concerns that are seen in autism, such as chronic constipation, sleep problems, abdominal distress. Next slide. This is just a kind of a mechanistic cartoon of where tyrosine, where alpha-methylparatyrosine hits. As you can see here in the drawing, it really hits at the early stages of conversion of tyrosine to dopa, which then goes on to impact the production of dopamine, norepinephrine, and epinephrine. Tyrosine hydroxylase, as you recall, is the rate limiting step. Is part of the feedback loop in terms of controlling the production of catecholamines. The reason that I think the inventor thought about using this compound was because of the fact that catecholamines are known to play a role of many neurotransmitters in regulating the dysregulated functions in ASD. In particular, many of us in the field know that dopamine receptor blockers, beta blockers, alpha-2 agonists do have some impact on improving some of the behaviors noted in autism. Sometimes those improvements do lead to improvements in socialization, but it's very variable. And we also know that we're not hitting the entire catecholaminergic, we're not modulating the whole catecholaminergic system when we use these drugs. We're kind of hitting various components. So the thought here is maybe rather than hitting specific receptors, which are associated too with side effects, just down-regulating the overall tone of the catecholaminergic system might be a benefit. And that was the basis for this kind of broad spectrum approach. Next slide. So an open label study was done with 8 children. This is published data. Open label, all the caveats and disadvantages of open label being obvious. Sorry. The caveats of open label, limitations being obvious and mentioned earlier by our previous speaker. Nonetheless, we saw really a strong, a substantive majority, anywhere between 6 to 7 of the 8 patients who showed improvement in multiple assessment measures. In the ADOS, for instance, we saw anywhere from essentially 5 of the 8 patients showed a significant drop in their scores, some dropping to below the cutoff for autism on the ADOS. The clinical global impression assessment of severity also dropped by usually about 2 points, occasionally 1 point. And then the Conners parent rating scales also showed nice trends in the positive direction for multiple domains within the Conners forms. Next slide. So with this, the original team before we joined decided to do a 40 patient safety and dosing range study looking at 100 milligrams 3 times a day versus 200 milligrams 3 times a day in a double-blinded randomized fashion. Essentially what they showed was the drug was safe and well tolerated, which was the primary outcome measure, and a better response seen at the total daily dose of 600 milligrams or 200 milligrams TID compared to 100 milligrams TID. None of the efficacy measures were powered for statistical significance. And to be honest, only 10 patients received active treatment in each of the arms. So extremely small numbers. We were not, again, looking for a statistically significant, but just kind of quantitative trends in the right direction. And in fact, we did see multiple measures, including the Vineland Adaptive Behavior Scales in the socialization domain, the ADOS-2, the CGI, repetitive behavior scales, and the SRS showed greater trends in the treated over the non-treated for benefit in the patients treated for only 28 days. So this was a 28-day study, thereby demonstrating non-statistical but nonetheless clinically significant differences. Based on this information, the FDA granted us fast track, as Chuck mentioned. Next slide. This is the overall flow diagram of the study. You can see this was because it was a safety study and there was not a lot of, there was no preclinical data available, but because the L enantiomer of this drug had been approved previously, the FDA allowed us to go in with the racemic mixture as long as we did 5 patients open label in each of the dosing cohorts first, check for EKGs and labs, make sure there were no adverse events, and then allowed us to randomize 15 patients initially in the 300 milligram daily dose, 100 milligrams TID, and then in the 200 milligrams TID, 600 milligram total daily dose. And so we randomized after the safety cohort was completed, 15 patients, 2-to-1 active to placebo, first in the 100 milligram cohort, and then in the 200 milligram cohort. In the active group in the second cohort, the 200 milligram TID cohort, we had 1 child withdraw due to a single seizure. That was not an SAE because it was just an out of hospital seizure. And then a few people, 2 people, 1 person withdrew in the placebo group and the high dose group just because they had a parent decided not to continue with the study. Next slide. And so what we saw here was on the left-hand panel, this was the change in clinical global of impression in the 2 groups. The placebo group showed a 0.3 point change, categorical change, and the absolute change in the treated group was almost a full point, 0.8, so about a 0.5 point difference. In the lower dose, we saw a lower change, a half a point in the treated group versus 0.2, but again, a dose response, a higher degree of change in the CGI severity in the high dose compared to the low dose. Similarly, in the ADOS, there was a mean full point change in the treated group compared to really no change in the placebo group at the high dose. And in the bottom right quarter panel, you can see a half a point change in the low dose compared to, again, no change in the placebo. Not statistically significant because of the small numbers and variability, but still an interesting dose response change that was observed. And again, some of these changes actually were seen across multiple measures, as you see in the next slide. We saw similar changes in the Vineland Adaptive Behavior Scales in the socialization domain. You can see a 6-point improvement, go back up, 6-point improvement in the Vineland socialization for the first 4 weeks, with then a return to baseline, a gradual return to baseline over 4 weeks over this ensuing 4-week period. And you see really virtually very minimal change in the placebo group. We also noted, did a kind of a responder analysis, and you can see that some of the response was driven in the high dose group by quite large changes in those who were treated, anywhere from 20 to 29 points amongst 2 out of the 7 patients that had a record. And then about 3-point changes in another individual, 3 points being kind of the minimum important clinical difference that we'd like to see with the Vineland. Next slide. On the SRS, we saw significant categorical drops from mild, in some cases from severe to moderate, from moderate to mild, and from moderate and severe to normal in those who were treated with the high dose. And the low dose, we really didn't see this level of change, but in the high dose, you can see on the motivation score, a 10-point drop compared to about 4 points on the placebo. Less difference, but still higher trends of dropping on the social communication T-score and the communication interaction T-score. Next slide. When we looked at the ABCs and the SRSs for particular areas where we saw responders being higher versus in the active group versus the placebo groups, you can see on the social withdrawal lethargy and on the inappropriate speech, we had patients responding on the active group about 40% and 50% respectively versus only 0 or 25% on the placebo patients. On the SRS awareness and social motivation, about 50% of the patients responded positively with a 2 or more point improvement. In social awareness, 60% for social motivation versus 20% and 0% respectively for the placebo patients. So when we did some sub-analyses, we saw much larger patients, much larger rates, between 40 and 60% response rates. Next slide. So we're actually just announced or in the beginning phases of initiating this study, which is a phase 2b crossover trial. We're starting off with 50 patients to randomize 40, or to complete 40, I should say, 50 patients randomized to complete 40. This will be total daily dosing of 400 to 600 milligrams, and it'll be BID dosing instead of TID dosing, and that's because we did some PK work in the interim between the last study and this study, and we're able to show a marked and prolonged half-life compared to the L enantiomer. So the L enantiomer of alpha-methylparathyroxine has a half-life of about 3 hours. When you use a racemic mixture, you extend that half-life to about 12 hours, making it doable to dose twice a day. And so this will be an AB-BA study where half the patients will receive drug first and then wash out and go to cross over to placebo, and the other half will receive placebo first, wash out, and then go to treatment. And that treated group will be split between 200 milligrams BID and 300 milligrams BID, 50-50. We're also stratifying, kind of a soft stratification around anxiety, because we do believe anxiety levels might be a modulator of responses. And our primary endpoint is the Vineland socialization domain, looking at actually the growth scale values as the primary measure, and then the standard scores. We'll also be looking at some key secondary measures, including the BOSC, the CGI weighted for socialization, and then also the PGI, which is a parent caregiver endpoint where the parents will choose the top 3 symptoms of concern and then rate those on the same 7-point Likert scale as the CGI. We'll also be using the social responsiveness scale, social responsiveness scales in the various domains within, and then the PRAS, the Parent Rated Anxiety Scale, and looking at the Aberrant Behavior Checklist subdomains, as well as the caregiver strain and some of the other domains, all the other domains actually within the Vineland, so the communication domain and the daily living, activities of daily living domains. Next slide. So this is just a kind of a listing of the outcome measures that we're using. I think these are pretty well known by most of the folks in this group, but key secondary measures being the BOSC. We're excited to be able to use the BOSC because it's starting to be an interesting endpoint to be used that gives some of that structured play activity scoring based off of some of the ADOS modules and methodologies that gives a sense of outcome that is measured with a third-party involvement, utilizing third-party scoring. And we're excited about that because, again, many of us have used the ADOS to look for changes, but of course it's not a tool that is very sensitive or meant to be used for long-term outcomes and longitudinal changes, whereas the BOSC is. So we're very excited to be using that. And then also switching to a GSV, growth scale value responder analysis, versus standard scores. Many people know standard scores can still decline in a child with autism, but it's really that rate of decline and whether or not you can stabilize and have new skills learned on the Vineland that is important. So even if a child, for instance, is flat on a socialization T-score, but grows or expands their abilities more than the typical child with autism, that is something to be really, I think, taken as a positive outcome and should be looked at. So the GSV allows us to do that. Next slide, Eugene. So we have 5 sites that have been chosen and qualified and are in various stages of starting. We have the Thompson Center in Missouri, the Thompson Center in Southern California, which I am actually a part of. The PI there is Dr. Gola. We have the Center for Autism, SARC, in Southwest Autism Research Center, the Center for Autism in at Rush in Chicago, and then the Wheel and Columbia CAD-B Center in New York with Jeremy Beenstra Vanderwall. So very excited to be running this, and we have 3 of the sites initiated already and waiting for the first patient enrolled, hopefully by this month. And I'll turn it over to Chuck.
Yeah, so the long-term plan for L179 is, as we mentioned, to get the results of the data in early 2023. We would expect a 3-year development program before we would get approval for the drug sometime in 2026, early 27. So that's our current target that we have. We, of course, will go to the FDA, and we're hoping the fast track designation can help speed up the process. We obviously will do a compassionate use program after this with good results for those patients that were in the trial. We will also look to do other work that the FDA has asked us to do, which we can get into at another point. The last slide just points out, Eugene, if you could hit the last slide there, the team that's been put together the last 5 years really to do this work that we have right now. You've met Tom. Dr. Tracy Fisher came to us with lots of experience in clinical development and regulatory affairs. She's leading the reg function for us. And Wynn is the senior director of our operations and is running our trial. Has a wealth of background. Tom used to work with her in previous roles. And Eugene is on the line now, our CFO. So we've got a small team. We would expect with positive results, we would grow the team another 25, 30 employees to help ramp up for the phase 3. So in essence, that's what we have. We'd like to turn it over for questions you may have that Tom or I can answer. Thank you very much.
Thanks, guys. Looks like Tom's addressing one of the questions that's come through via the chat, so thank you. One question I have you guys looked to see, given the dopamine theory of ADHD and what you guys are doing, have you guys seen, it doesn't look like there was much presented on changes in attention as a response to the intervention. Have you guys looked at that in any detail? Have you looked at subgroups of kids with autism with comorbid ADHD? Just, I'd love to hear your thoughts there.
Tom, go ahead.
Sorry, I was muted. We did, in the previous study, look at the Connors and in particular to see if there was any increases in hyperactivity. There really was no signal because we were worried about that as well. Is that, are we going to affect attention and focus? And did not see any worsening of that, across the 2 groups, but small numbers again. So we will be keeping track of that, especially with the ABC. So we'll have hyperactivity as a measure based on the variant behavior checklist.
Okay. All right. Well, I believe that's it in terms of the questions. Pramila, is it time for a break? Thank you, guys.
We will be taking a break till, I believe, till 3 o'clock. And then start with one more presentation in the industry talks and then move on to more basic science.
All right. Thank you.
Thank you, guys.
Everybody.
Thank you so much.